In the 1920s, Soviet biologist Ilya Ivanov tried to create a human-ape hybrid. He was already known for his work in artificial insemination, and had created hybrids between animals like horses and zebras. Backed by the Soviet government, his goal was to prove Darwin's theory of evolution, challenge religion, and possibly create a new kind of worker, strong, obedient, and easier to control. The government gave him funding, chimpanzees, and access to facilities in West Africa and the Soviet Union. Ivanov tried to inseminate female chimpanzees with human sperm, but none became pregnant. When that failed, he reversed the plan, trying to inseminate women with ape sperm. These experiments were based on a false belief that humans and apes were biologically close enough to reproduce. In reality, the genetic gap was too large, and none of the attempts worked. What made the project worse was how unethical it was. There are reports that some women were chosen without clear consent, and that prisoners may have been used as test subjects. These people likely had no idea what they were agreeing to, or were pressured into participating. The whole project treated humans as tools for state goals instead of as individuals with rights. Ivanov's career ended badly. In 1930, during Stalin's political purges, he was arrested and exiled to Kazakhstan, where he died a year later. His failure wasn't just scientific. It exposed how dangerous science can become when it's driven by ideology and done without basic respect for human life. The Nazi twin experiments were some of the most disturbing medical abuses in history. They were part of the Nazis' larger plan to promote racial purity and eliminate people they saw as weak or inferior. Starting in 1933, the regime had already forced sterilizations and killed thousands of disabled people. From May 1943, at Auschwitz, SS Dr. Josef Mengele took charge of cruel experiments on twins, mostly Jewish and Roma children. Mengele, who had trained as a medical researcher, used the twin studies to try and prove false ideas about genetic superiority. He ignored all medical ethics and used the children like test subjects in a lab. He often used one twin as a control and experimented on the other. These experiments included infecting children with diseases like typhus, injecting dyes into their eyes, giving unnecessary blood transfusions, and amputate limbs. In some cases, he even tried to sew twins together to create conjoined twins. Many children died from the procedures, and those who survived were often killed afterward, so their bodies could be dissected. Out of around 1,500 sets of twins, only about 200 children survived. The Nazi doctors saw these children not as people but as objects for testing. Although Mengele escaped justice, the horror of these experiments shocked the world and helped bring an end to eugenics as a science. More importantly, they led to the creation of the Nuremberg Code, which set the rule that no one can be used in medical research without full informed consent. CRISPR gene editing in human embryos is a major scientific breakthrough that could one day stop inherited diseases before a child is even born. It works by using a tool, based on a system found in bacteria, that can cut and change DNA at specific spots. This offers a way to fix genetic problems early, possibly preventing serious illnesses like Huntington's disease or cystic fibrosis. But using CRISPR on human embryos comes with serious risks and is not allowed in most countries. One major issue is that CRISPR can sometimes cut the wrong part of the DNA, causing new problems. Another problem is mosaicism, where only some cells in the embryo get edited while others don't. Also, current tests aren't always good at checking if the changes worked correctly or safely. The body's natural DNA repair process is hard to control, and sometimes it causes more harm than good, which makes the results very unpredictable. There are also big ethical concerns. Edits made to embryos affect not just one person but all future generations, and those people can't give consent. Some fear the technology might be used to create designer babies for traits like intelligence or appearance, which could widen the gap between rich and poor. Although some countries have strict rules against this, others don't, raising fears that people might travel to avoid restriction. As the science moves forward, clear rules, better safety tools, and honest public discussions will be key to deciding how this powerful technology is used. The glow-in-the-dark rabbit, named Alba, was created in 2000 by artist Eduardo Cac as part of a project meant to spark public conversation about genetic engineering. Alba was born in a lab in France and was genetically modified to glow bright green under blue light. This effect came from adding a special gene called EGFP, which originally came from jellyfish. Scientists used a method called zygote microinjection to insert the gene into her DNA before she was born. Cac wasn't just interested in science. He wanted to challenge how people think about life, nature, and the role of humans in changing other living things. Alba was meant to be both a living creature and a piece of art. Her glowing fur made her famous, but also caused a lot of controversy. Many people questioned whether it was right to change an animal's DNA just for an art project. Critics said it raised issues about animal rights, scientific limits, and the use of living things for human goals. Even though Alba never lived with Cac as planned because the lab refused to release her, she became a symbol in debates about science and art. Her story appeared in pop culture shows like The Simpsons and Sherlock, and inspired books by authors like Margaret Atwood. The Free Alba campaign gained attention worldwide, highlighting the tension between scientific progress, artistic freedom, and animal welfare. The spider-goat hybrid is a real scientific breakthrough, where goats are genetically modified to produce spider silk proteins in their milk. Scientists did this by inserting a spider silk-producing gene into the goat's DNA, so when the goat makes milk, it also makes the silk protein. This idea came about because farming spiders isn't practical. Spiders are territorial and often eat each other, making it nearly impossible to collect silk in large amounts. Using goats solves that problem, since their milk can produce a steady and large supply of the protein. This research was partly backed by the U.S. military in the 1990s, and later advanced by Dr. Randy Lewis at Utah State University. The spider silk protein made from goat milk is valuable because it's incredibly strong, about 5 times stronger than steel of the same thickness, and also very stretchy, light, and safe to use inside the human body. Because of these qualities, it can be used in making things like bulletproof armor, super strong fishing line, and medical items like stitches, wound coverings, and structures to help regrow bones or nerves. Even though it's a big step forward, some people worry about the ethics of changing animals just to serve human needs. These concerns raise questions about how far we should go with genetic engineering. Today, scientists are looking for better ways to produce spider silk using bacteria, yeast, or genetically modified silkworms, which might make production cheaper and more scalable. The Vacanti mouse was part of a well-known tissue engineering experiment in the 1990s, led by Charles Vacanti, Robert Langer, and Joseph Upton. In this study, scientists grew an ear-shaped piece of cartilage on the back of a hairless mouse. They didn't actually grow a human ear, it just looked like one. The process started with a plastic-like scaffold shaped like an ear. This scaffold was filled with cartilage cells from a cow, then placed under the skin of a special kind of lab mouse called a nude mouse. These mice have very weak immune systems, so they don't reject foreign cells like regular mice would. Because of this, the ear-shaped cartilage could grow without being attacked by the mouse's body. The purpose of the experiment was to show that scientists could grow tissue in a specific shape using a mix of living cells and man-made structures. The cartilage that grew wasn't a working ear. It had no nerves or hearing function, but it was a big step in learning how to build body parts in the lab. When photos of the mouse with the fake ear went public, they sparked both excitement and concern. Some people saw it as amazing progress, while others felt uneasy seeing a living animal altered in such a way. Still, the experiment helped push forward research in growing tissues and organs for future medical use. Human-monkey chimera embryos are early-stage embryos made by combining human and monkey cells. Scientists create them by inserting human stem cells, special cells that can turn into any type of tissue, into monkey embryos. This is done to study how human cells grow and interact inside another species, especially one closely related to us. The main goal is to learn more about early human development and possibly find new ways to grow human organs for people who need transplants. Because monkeys are closely related to humans, their embryos provide a useful model for research. By watching how human cells behave inside the embryo, researchers hope to better understand diseases and how tissues form. This could lead to future breakthroughs in medicine, especially in creating custom organs from a person's own cells. However, this kind of research raises big ethical concerns. People worry about what could happen if too many human cells are added, especially in areas like the brain, which could affect the animal's behavior or mental abilities. To prevent this, strict rules are followed. Embryos are usually only allowed to grow for a limited time, often just a few weeks, and are never implanted to grow into full animals. Even with limits, these studies offer valuable insight into how cells from different species work together and how we might one day grow healthy human organs in animals. It's a careful balance between scientific progress and ethical responsibility. The de-extinction of the dire wolf is a new area of genetic research, focused on bringing back a large predator that went extinct about 10,000 years ago. The dire wolf was not just a bigger version of the gray wolf. It was a completely separate species with a stronger build, a larger head, and more powerful jaws, designed to hunt large prey during the Ice Age. Scientists are now using ancient DNA recovered from dire wolf fossils to study its genetic makeup and compare it with living relatives like gray wolves and dogs. Because the DNA is so old and damaged, it's not possible to clone a dire wolf directly. Instead, researchers are identifying specific genes that made dire wolves unique and using tools like CRISPR to insert those genes into the DNA of modern animals. The goal is to eventually create an animal that closely resembles a dire wolf. This work could help scientists understand how extinct animals lived and how they might fit into today's ecosystems. It also raises major ethical and ecological questions, like whether it's right to create an animal that's been gone for thousands of years, or how such an animal would affect current wildlife. Some experts also worry this research takes attention and funding away from protecting species that are endangered right now. Still, the dire wolf project is one of the most advanced examples of how science is trying to reverse extinction using gene editing and ancient DNA. Thanks so much for watching. If today's video sparked your curiosity or made you think a little deeper about science, ethics, or the future of our world, consider hitting that like button and subscribing for more content just like this. Also, if you have any video ideas, please feel free to let me know in the comments, as I read every single suggestion. Enjoy the rest of your day.